by different arrangements of the exons as the exons are being spliced out of the pre-mRNA. And so that's one reason why they think we have introns, is to modularize the protein encoding segments to allow alternative splicing. There's another reason why they think we might have exons and introns in our genes, and that is to help create new proteins. For a species to evolve, it has to generate new proteins, so new genes. And the modularized nature of genes, like you see here, may facilitate the evolution of new genes. Here's how this can happen. Here's one way this can happen. And to understand this, we have to go back to meiosis 1. Remember that in prophase of meiosis 1, there's a phenomenon that goes on called crossing over between homologous chromosomes. Let's say this is your maternal chromosome number 1, and this is your paternal chromosome number 1. In prophase of meiosis 1, they line up with each other. And then segments of one can break off and exchange with a segment of the other. It's kind of a random process in regards to which segment breaks off and exchanges with the other, but it can happen, like you see there. Now, notice from this cartoon here, equal segments are crossing over. That the piece that broke off here and went down there is exactly equal to the piece that broke off there and went up there. And so as long as there's equal crossing over, each chromosome is not going to gain or lose any genes, because these are homologous chromosomes. They're carrying the same genes in the same order. And so whatever genes were sent down here, it's going to pick up those same genes, so to speak, from this other homologous chromosome and get up there, as long as there's equal crossing over. Occasionally, very rarely, but occasionally it does happen, there's something called unequal crossing over. In unequal crossing over, go back, the pieces that cross over are not exactly the same. Imagine on this homologous chromosome, we have gene A and B, and on this homologous chromosome, we have those same two genes, of course. But let's say the breakpoints look like this. So the segment that's going down to this paternal chromosome is not the same one that's going up to that maternal chromosome. Like that. So notice that you create some Franken genes. That's not the official term, but I'm saying that you're ending up with a gene, a new gene that's created by splicing certain parts of one gene to different parts of another gene. And likewise down here, certain parts of this gene have now been combined with certain parts of this other gene over there. Well, thanks to the genes being modularized, where you have these exons spaced apart from each other by the introns, you'll have you're not going to disrupt the entire structure of the gene. Or what I mean to say by that is you'll have some good functional exons here and some good functional exons here, but now are intermixed with each other. And so you can get a protein that combines functions from two other proteins. In other words, you might have a binding domain here for hormone X being spliced to, I don't know, an enzyme for carrying out oxidation of beta-lactamase or something like that. It's a new function that's never existed before, because you're mixing together two genes. Or maybe over here, you might have an enzymatic binding site for ATP being combined with a chloride ion pumping protein. So now you have an ATP-powered chloride ion pump that you didn't have before. So by dividing the gene into exons, it facilitates their ability to mix and match by unequal crossing over, and this may help us evolve new and improved proteins. It's a theory, but it kind of makes sense. All right, well, to finish off this chapter, I thought I would go over the complete structure of a fully processed RNA. That's not the picture I was looking at. Here we go. All right, so when the primary transcript is rolled right off the gene, it's that light blue one right there. So it's initially as long as the gene, but it needs to be processed, and one of the ways it has to be processed, of course, is that this 5' cap has to be attached to one end, and the introns have to be cut out, and the poly A tail has to be added to the 3' end. But once it's processed, it's now referred to as the mature mRNA. That exits the nucleus, and then out there in the cytoplasm, the ribosome binds to it, and the tRNA shuttle in amino acids, and the codons get read, and the protein gets made. But what I want to do is focus in on the mature mRNA itself, and let's review all the parts that it has. All right, now at the extreme 5' end, you have this thing called the 5' cap, remember, and that's a modified GTP nucleotide. There's guanosine triphosphate, and that's different from the normal Gs that you find in an mRNA, because most of the Gs and As and Us and Cs only have one phosphate. This one has 3 on it. Why is it there? Well, it does several things. Remember, it helps the mRNA exit the nucleus. It protects the mRNA from being degraded by enzymes in the cytoplasm, and it helps the ribosome find the messenger RNA, bind to the messenger RNA. After the 5' cap, you start having the part of the messenger RNA, which is the exon spliced together. So I'm showing this as little boxes spliced together to remind us that the introns have been cut out. And so everything you get in mature mRNA is exons. In the first exon somewhere, you're going to find the start codon. I think we're now going to zoom in on that. There it is. And the start codon is always an AUG. Maybe I should describe it as the first codon is always an AUG. And that encodes the amino acid methionine. There it is. So all proteins begin with methionine as their first amino acid, because that is always codon number 1. But notice that the first codon, the first AUG, is not the first nucleotide of the mature mRNA. There's always going to be some nucleotides upstream of that first codon. Those are called the 5' untranslated region, because the ribosome does not translate them into any amino acids. The 5' UTR. Now, I think I mentioned this before. You might wonder, well, how does the ribosome know not to make any amino acids out of these? For instance, here's a CCG. Why is it that the ribosome doesn't see that and say, oh, CCG, that encodes proline. I think I'll start this protein with a proline amino acid. Well, the ribosome knows that it's supposed to scan along the 5' UTR until it finds the first AUG. That's just part of the programming of the ribosome. So it basically ignores everything that it finds as it scans along until it comes to the first AUG, and then it says, aha, that is where I'm going to start actually building the protein from. All right, so there's our start codon, and there's codon number 2, codon 3, and codon 4. So the ribosome is just going to keep reading all the following codons after codon number 1 all along the mRNA. And so, here's exon number 2. It'll have its own codons within it, and the ribosome will read those. Then exon number 3 and exon number 4 with more codons and more codons and more codons. The last exon, let's look at this one right down here. The last exon is going to have the last codons of the gene. And remember, the very last codon is always a stop codon, which is not translated into any amino acid. It's just a message to the ribosome to stop making amino acids, that its job is done. And there are 3 stop codons. Let's see. Oh yeah, so here is the last, there's the last exon. And there you see the stop codon right there, the UGA. But notice, again, that the stop codon is not the very end of the messenger RNA. There's still going to be some more nucleotides downstream of that. That zone is called the 3' untranslated region. Again, untranslated because no amino acids are going to be made from it. The ribosome knows to stop making amino acids when it hits the stop codon. Within the 3' untranslated region, you're always going to find what's called the polyadenylation signal, AAU, AAA. And the polyadenylation signal, you remember, is used by an enzyme in the nucleus. There's an enzyme in the nucleus that looks for the polyadenylation signal in the 3' untranslated region. And then a few nucleotides downstream of it, this enzyme adds a bunch of A nucleotides, a bunch of AAAs, which are right there. And that's called the poly-A tail. The poly-A tail serves similar functions to the 5' cap. The poly-A tail helps the mature mRNA exit the nucleus. It helps protect it from degrading enzymes in the cytoplasm, and it also helps the ribosome find and bind on to the messenger RNA. But notice that the sequence of A's was added after transcription, and so you don't find this long series of A's in the actual gene that encodes this mRNA. They are added after the fact. All right, any questions on the messenger RNAs or their splicing? Okay, just as a preview, today in laboratory, you're going to be going over, doing an exercise involving everything we talked about today. You're going to be given a paper version of a gene, maybe something similar to this, but of course it's going to be G's, A's, T's, and C's. And then you're going to find the promoter sequences, and then laying out a piece of paper next to your gene, you're going to transcribe from the template strand and make a messenger RNA, and then you're going to play spliceosome and cut out the introns and then try to make a protein out of it. Kind of like when you guys were the chromosomes going through a drill for mitosis, meiosis, same thing, but now it's going to be transcription and RNA processing and translation. All right, well, let's jump into the next chapter then. So we're going to do viruses and prokaryotes now, but do have the DNA technology chapter printed up and carry it with you at all times. Well, don't bring it into the shower, but everywhere else carry it with you. Okay, well, let's see. So the prokaryotes are the species that are in the bacteria kingdom and the species that are in the archaea kingdom. And remember, those are unicellular prokaryotic organisms. And the very definition of a prokaryote is doesn't have a nucleus. But I guess I haven't talked too much about viruses at this point, so maybe before we do the prokaryotes, let's talk about viruses. As you can see from the diagram here, viruses are even smaller than prokaryotic cells. And prokaryotic cells are pretty darn small compared to our big eukaryotic cells. So viruses are pretty darn tiny. Viruses are extremely simple organisms. So simple, in fact, that not all biologists consider them to actually be living organisms. I do, but a lot of books just say no, that they're not technically alive. Because they don't meet some of the general characteristics that we associate with living things. Viruses do not have any metabolism. They do not have any active enzymes inside themselves for digesting glucose or getting energy for themselves or anything like that. They don't have any organelles, and this is really telling, they don't have any way of reproducing themselves. To reproduce them, the only way a virus can reproduce itself is by getting inside a living cell, sometimes called the host cell, and tricking the cell into reproducing the virus. Oh, I guess before I mention that, I was going to talk about the diseases. We often associate bacteria and viruses with diseases, and bacteria don't always cause diseases. There's a lot of actually beneficial bacteria in the world who wouldn't hurt a fly. Some bacteria can definitely cause diseases, but a lot of them do not. But viruses are always disease